Assalamualaikum warahmatullahi wabarakatuh, and hello all. We are UiTM students from course AS249, and we would like to share our experiences during our field trip for our ecology subject BIO464. Our team consists of two members, me, Faiz, and my teammate Alif. I will discuss the objective of our visit as well as itinerary, whereas Alif will answer the practical question that follow. Without further ado, let's begin with an overview of this whole project. This field trip project in general aims to bring students closer to nature and to allow students to appreciate the natural wonders of Earth. At the same time, the activities conducted during the visit allow students to learn how to population samplings work, as well as to give students first-hand experience of working at the field as ecologists. The field trip was split into two sections, one in the morning and another in the afternoon. We will begin with our trip to Sijangkang Forest Reserve, which took place in the morning. Formerly known as the Sijangkang Mangrove Recreational Park, it was developed from an illegal dumping site. It spans 5.5 km in area and has been open to the public since 2015. The village folk have done a great job at maintaining the scenery since, and many schools and companies have visited the place to plant mangrove. We arrived at 8 o'clock via bus. The journey was scenic as we rode through forest and palm plantation. We got down and had a lovely breakfast accompanied by the cold morning air. Students from the UiTM also arrived at the scene. The opening ceremony was conducted around 9 o'clock, and we had a quick aerobic exercise session before gearing up and entering the mangrove forest. We were guided by the camp managers to the site. I will admit no one was safe from mosquito bites. After walking along wooden bridge through the shady trees, we finally arrived at the site. All of us descended into the mud and began digging holes for the saplings. We enjoyed our time and took many photos of ourselves struggling through the thick mud and handling the heavy tools. Some handled it better than others, but we made sure everyone planted at least one mangrove sapling. The mangrove sapling must be planted upright and spaced evenly to ensure its proper growth later on. After the sweaty and grueling work, we walked back to the camp for the refreshment and lunch. We were covered in mud and mosquito bites, but the work was rewarding. Most of us fell asleep during the bus ride to our next stop, Putrajaya Botanical Gardens. Located in Precinct 1, Putrajaya Botanical Gardens is home to both rare plant species and animal conservatories. It spans 93 hectares in area and has been open to the public since 2001. The place functions as a way to educate the public about rare species of flora and fauna in Malaysia and other parts of Southeast Asia, while also becoming a conservatory for 6 species. We arrived at 2 o'clock. We did our prayers there before assembling at the reception hall. We were greeted by the managers of the institution. They were welcoming and very friendly with us. Immediately as we began our trek into the gardens, our guide spared no effort at explaining the various different species found there. We were definitely surprised at how diverse the flora and fauna is in Malaysia, especially of how there actually are species endemic to our country there. One of them is the plant known as daun payung or Aracasia johannestegimania magnifica. It only grows in forest slope in Peninsula Malaysia. We went all around the gardens via go-kart. The air was windy and the sky was cloudy, which made it pleasant to look at the various trees and gardens. We tasted a lot of exotic fruits too, such as kirkap, rukam and water apples. Finally, at 4 o'clock, we ended our tour and got to completing our lab reports for ecology. We wrapped up our job at 6 o'clock and headed back to our dorms. I will leave the matters of the practical work to my teammate, Alif.
Thank you, Faiz, for presenting the introduction as well as the itinerary of our field trip to Sijangkang Forest Reserve and Putrajaya Botanical Garden. Now, I would like to present the practical work that we did in Putrajaya Botanical Garden. There are 2 practical works that we did. The first one is in regards to basic concepts of statistics and variation in leaves. And the second one is the factors that affect the distribution and abundance of organisms in a quadrant. We will first begin with practical 1. The objectives of practical 1 is to understand and describe what does the mean, variance, and the standard deviation mean, to understand the concept of correlation, and to determine the relationship between 2 variables. For practical 1, we were required to measure the lengths and widths of leaves on the ground. 3 sets of 10 leaves were picked up and their lengths and widths were measured. From there, we were able to derive the mean, variance, and standard deviation of the lengths and widths of each set respectively. The results were tabulated in Table 1. These are the results. All values in Table 1 are recorded in centimeters. It is shown that among the 3 sets, there is a significant difference of mean, variance, and standard deviation of the length and width respectively, despite all leaves having roughly the same size. We will move on to discussion questions. Question 1 is, discuss the differences or similarity of the mean of length between different sets of leaves. Do the same for the mean of width. At a glance, the leaf lengths from all 3 samples have a greater variance compared to its width. The difference in average length of the shortest and the longest totals to 0.75 centimeters, whereas the difference in average width of the shortest and longest totals to 0.18 centimeters. Therefore, among the 3 sets of leaves, they are more similar in width than in length. However, the differences are otherwise within the length of 1 centimeter, suggesting that these leaves all came from the same tree, or different trees of the same species and similar environments. Question 2. What is meant by variance? Variance is how far or different the sample values are from the mean of a set of data. In our case, variance is how different the length and width of the leaves are from the average of the set. We have found that the variance of length varies from 3.7 centimeters to 2.2 centimeters, whereas the variance of width is from 0.2 centimeters to 0.4 centimeters. Then, what is meant by standard deviation? Standard deviation is how spread the sample's data is relative to the mean. The closer the standard deviation of data is to absolute zero, the closer the majority of data values are to the mean. In our case, a lower standard deviation means the leaves have very little size differences, and vice versa. We have recorded that the standard deviation of the leaf length is within 2 centimeters of difference, whereas the standard deviation of the leaf width is within 0.5 centimeters of difference. To follow, the leaves have different measurements of width and length. What is the meaning of this statement? Discuss it with respect to variance. This statement means that the leaves do not follow a rigid size guide, instead follow a natural variance of sizes. Some are bigger than others, some are long and thin, and others short and wide. This variance would require different measurements in order to quantify the leaf size data accurately. Question 3. Why is it necessary to have different sets of measurements for the measurement of leaf width and length? There are 3 reasons why. Firstly, The different measurement systems are much easier to apply in practice compared to a surface area approach. A surface area approach would require the use of computational imagery, and that would take up energy, space, and time. Measuring length and width only requires a ruler, and its measurements are straightforward, which save processing time and allow for more samples to be collected. This is especially useful in the field. Number 2. Having 2 scalars instead of 1 also accounts for species differences, as leaves from different species grow differently and have different length to width ratios. So, depending on the age of the leaf, some leaves might be more broader and some leaves might grow more thinner with age. This is important to know and is why there are 2 different sets of measurements. Thirdly, changes in leaf width and length are caused by a myriad of biotic and abiotic factors. Knowing exactly how much changes as well as recording patterns allows for ecologists to better record changes and trends in the environment based on the relation between leaf width and length over time. Now that is practical 1 completed. Next we will move on to practical 2, which is investigating the factors that affect the distribution and abundance of organisms. The objectives of practical 2 are to introduce the concept of quadrants and teach students how it is used to estimate distribution and abundance, to enhance the observation skills of students and improve their recording skills, and thirdly, to infer from observations the possible factors that affect the distribution and abundance of organisms. For this practical, we were required to section off a 10-foot by 10-foot area and count the amount of organisms that were present inside. We did 2 quadrants in total, and we tabulated the results. The results were marked out in the form of a drawing where different species of trees were given different symbols. The results are as shows. The following is the map of Quadrant 1. In Quadrant 1, we had found 3 individuals of Dilenia subfruticosa, 1 individual of Dilenia excelsa, 1 individual of Hopia odoranta, and 1 individual of Calophyllum inoculum. In Site 2, we found 1 individual of Paritia insignis, 1 individual of Terminalia catapa, and 1 individual of Liquala petata. We now move on to the discussions. Question 1. Define the term quadrant in practice. In practice, quadrants are a square area chosen to sample and observe the types of trees distributed in an area, as well as to measure the abundance, diversity, and composition of the tree species present. We used a 10 square feet quadrant for our measurements. Question 2. What are 2 basic requirements of all quadrant techniques? They are knowing the area of the quadrant and accounting for all members of the population in the quadrant measured. Question 3. What would be the advantages and disadvantages of using a larger/smaller quadrant to survey sedentary organisms? For a larger quadrant, it is better used because it has a better sampling range and increased accuracy of overall density measurement due to its large area. However, it is time-consuming to set up and count every single organism inside the quadrant. Not only that, it is not very precise at determining the edges of specific biomes and ranges of the species. On the other hand, a smaller quadrant is more precise at defining the true borders of the species range. However, it can miss crucial species in the process and requires setting up many times in order to achieve the same effects as one large quadrant. Question 4. Problems with spatial scale often make it difficult to directly estimate average densities in habitats such as large forested areas or in aquatic habitats where one cannot directly see the animals or plants of interest. Explain the issues that concern ecologists in conducting the sampling method. There are 4 issues. Firstly, whether the size of the quadrant is large enough to include enough species. Secondly, whether the number of quadrants is enough to account for locational variance of the species population. Thirdly, whether all members of the population have been accounted for in a quadrant. And finally, whether the location of the quadrant sample has the least deviation from the overall population density of that specific location. Question 5. Why is there difference in the number of plant species in a quadrant? Hypothesize or make your own assumption. Discuss the factors that would have caused this type of distribution. We have concluded that there is not only a difference in number of plant species in each quadrant, but also completely different plant species found, despite the quadrants that we took being within 50 meters from each other. This can be attributed to the quadrant size being too small and too little quadrant samples being taken. We only took 2 samples, where for a place as big as Putrajaya Botanical Gardens, it will be much better to take 10 to get a much better estimate of the population density. Because we took very few quadrants, this reduces the number of plant species found among all quadrants. However, this does not explain why both quadrants have different plant species found despite being close together. The explanation for this is due to the nature of our sampling environment. The sampling is done in a man-made forest where the location of each plant is deliberately. This allows for a very high species diversity within a very small sample area. In a natural forest, there will be more members of the same species clustered near each other. That is Practical 2 completed. In conclusion, this double field trip to Sijangkang Forest Reserve and Putrajaya Botanical Gardens has been very beneficial to students taking ecology. It allows us to experience firsthand what working in the field is like, and it also promotes our observational and critical thinking skills. The quadrant activity teaches us the crucial skill of recording our observations and making inferences from data. The mangrove plantation activity serves as a reminder that healing and caring for the planet is a global effort and is our responsibility as humans. Both activities have also shown us how diverse nature is and why we should strive to learn more about it. Future students taking ecology should attend this double field trip to broaden their horizons and put into context what they have learned in class. Hence, that is all from us, Team Faiz and Team Alif. Thank you very much for watching. Assalamualaikum warahmatullahi ta'ala wabarakatuh.